Good morning, students. Today we are going to see about the food additives. So it starts the class with the food additives definition. So food additives are the substances that are intentionally added to the food to enhance the taste, flavor, and texture. So, which is what is a food additives? It is been intentionally added to the food, either to enhance the taste, flavor, or a texture. So there are some examples of the food additives. They are like gums, emulsifiers, preservatives, flavor, and color. And nowadays the consumer are preferring a more for the natural products, over the chemicals, because the chemicals products which has a various ill effects to the body. So the biotechnology is an important process to produce all the natural products. So first we are going to see about the acetic acid. Acetic acid, the chemical formula of the acetic acid is CH3COOH, principal constituent of the vinegar. So literally this acetic acid is being translated as a sour wine. So one of the oldest product of fermentation which is used by the man. So the microorganism which is used for the production of the acetic acid is Acidobacter acetate, which is normally used to produce a vinegar which makes up to 14% of the acetic acid. And when cider, wine, beer, and barley malt is used as a starting material, we get about the 5% of acetic acid. And the color and the taste of the vinegar, which depends upon the source. What are the source we are going to use for the production and the method of the production. These two which gives the color and taste of the vinegar. And traditionally, the vinegar was being produced in a wooden cask, which has been filled with a wooden shaving in which the wine has been sprayed. You can see it in the picture. The first picture, this is called as a wooden cask, where it is being filled with the wooden shavings, and there this wine has been sprayed there, and it is kept for the fermentation. Once the vinegar is formed after the fermentation process, the vinegar doesn't have any color, and after that it will get filtered and it is being used for the cooking purposes. And the table vinegar, what is a table vinegar? The vinegar which is used for the cooking purposes, they are called as a table vinegar. They contain about 4% to 8% of acetic acid, and that is used as a condiment. And there's also used in the pickling of vegetables and other foodstuffs. When it is used as in the commercial food pickling process, the concentration will get vary, where we use a more concentrated solution. The basic requirement for the vinegar production is a raw material that will undergo an alcoholic fermentation. Usually we use like a apple, pears, grapes, honey, syrup, cereals, and hydrolyzed starches, and also the beer and wine are also used, and it is an ideal substrate for the production of the vinegar. The best raw material which is used in Europe and United States is a are cider and vinegar. And to produce a high quality product, it is essential to have a good raw material which is mature, clean, and in good condition, so that we get a good quality vinegar. So let's see about the principles of acetic acid fermentation. So vinegar can be made from any type of fermentable carbohydrate source. For example, like fruits, vegetables, syrups, and wine, anything we can use that has a carbohydrate. So whatever the raw material we are going to use, the fermentation process will be the same. So the production of vinegar depends upon the mixed fermentation, which means that involve both the yeast and bacteria. Without the one, the fermentation process is not completed. The fermentation is usually initiated by the yeast. So the yeast which helps to break down the glucose into an ethyl alcohol with the liberation of carbon dioxide gas. Following that, bacteria has been used, so which helps to oxidize the alcohol into acetic acid and water. So we can see the equation. So glucose, the yeast reaction, where the glucose yeast which reacts with the glucose and forms the ethyl alcohol and the liberation of carbon dioxide. Next comes the bacterial reaction, where the alcohol is being converted into an acetic acid and water. So oxygen is also is required for the conversion of the acetaldehyde to acetic acid. In general, the yield of the acetic acid from glucose is approximately 60%, where that is 3 parts of glucose yield 2 parts of acetic acid. So from the 3 parts of glucose, we get what? 2 parts of acetic acid. So microorganisms, what are the microorganisms that are involved? The organism involved in the vinegar production, which needs a grow at the top of the substrate, and they will be like a jelly like mass, and that is called as a mother of vinegar. And that mother, they contain both the acidobacter and yeast, that is both the bacteria and the yeast. In bacteria, the microorganism which is we use is acidobacter acetate, and in yeast which is a Saccharomyces cerevisiae. And it is also important to maintain the acidic environment to suppress the growth of the undesirable microorganism and to increase the presence of the acetic acid. So what is the percentage we get? It is a 10 to 25% by volume we get. After the fermentation process is complete, we get 10 to 25 percentage of the vinegar. Okay students, next us we will see about the citric acid fermentation.